So now we have specification number 5, which asks us to explain why observed ratios often differ from expected ratios, especially when there are small numbers of offspring. Here, it is important to understand that Punnett squares only show the expected or predicted ratios based on probability, not the exact results that will always occur in real life. Observed ratios do not always exactly match expected ratios because genetic inheritance is based on probability, not certainty. Ratios like 3:1 are predictions based on chance, assuming that each fertilization event happens randomly. Each offspring has an equal chance of receiving either allele, but it is not possible to control which gamete will combine with another. During fertilization, gametes combine randomly, and each event is independent. This means the outcome of one fertilization does not affect another, so results can vary simply due to chance. The effect of small sample size is very important. When only a few offspring are produced, chance has a greater influence on the results. For example, instead of getting 3 black and 1 brown, you might get 4 black or 2 black and 2 brown. Even small differences in numbers can change the ratio noticeably. Expected ratios such as 3:1 or 1:1 are based on calculations using Punnett squares and assume ideal conditions with a large number of offspring. As the number of offspring increases, the observed results tend to get closer to the expected ratios because random variations even out over time. For example, a much larger group is more likely to show a ratio close to 3:1. Other factors can also influence the results. Environmental conditions may affect survival, and errors in observation or recording can lead to differences between expected and observed ratios. Now here, this example clearly shows how sample size affects the difference between observed and expected ratios in genetic crosses. In a small sample size, such as 8 offspring, the observed results can vary a lot from the expected ratio. For example, some groups may show 5 purple and 3 white, others may show 7 purple and 1 white, or even all purple with no white at all. These results are very different from the expected 3:1 ratio. This happens because of chance, where random fertilization can strongly affect the outcome when only a few offspring are produced. Even 1 or 2 different outcomes can change the ratio significantly. The expected ratio, based on a cross between 2 heterozygous individuals, is 3 purple to 1 white. This prediction comes from the Punnett square and assumes ideal conditions with many offspring. In contrast, when the sample size is large, such as 200 offspring, the observed ratio becomes much closer to the expected ratio. For example, a result like 148 purple and 52 white is very close to the predicted 3:1 ratio. In larger samples, the effects of chance become smaller because variations balance out over many individuals. This shows that probability is a prediction, not a guarantee. Small samples can give unpredictable results, while large samples tend to match the expected genetic ratios more closely. The key idea to remember is that as the number of offspring increases, the difference between observed and expected ratios decreases, and the results become more accurate.